technologies are all actually working quite well. I mean, some XEMS has some nice properties to them that allow it to work reasonably well in many different scenarios, as does Mate, and you really can't go wrong with the technologies these days as they're getting better and better. For me, it's how we're developing these technologies to answer specific biological questions, and I ultimately think it's the technologies and how they're being developed to answer these biological questions that is most relevant. As a technologist, there's always the danger of developing the technologies for the sake of developing them, and that's something that we've always tried to be careful of not doing. So I have a list of some of the more recent papers that have come out of our lab. The most, at least for me, the most significant one is the Nature Biotech, that paper that just came out a couple of weeks ago. And for those of you who are interested, you should check this out because it's ultimately the general theme behind my overall presentation today. I'm going to cover a lot of different topics on the mass spectrometry side, which is the technology of choice that we use. I'm going to focus a little bit on that. Also, the informatics that we developed with respect to XEMS. But the ultimate goal of what I'm trying to, the excitement I'm trying to generate in metabolomics is not in the technology, but how we're applying it. And I'm going to be covering quite a few different topics today, but I'd ultimately like to say that it's going to come down to a fairly simple message at the end. So even though I'm going to be covering lots of different things, please keep in mind that to some extent, it's all going to get wrapped up together at the very end. For those of you not that familiar with XEMS, it's in its simplest form. Well, it does something relatively straightforward. It picks peaks, it aligns the peaks, and ultimately allows us to statistically pull out the most interesting molecules. Currently, it has on the order of a little over 24,000 registered users. And I think one of the reasons why it's so widely used is that it's freely available. And what's I think also very important is that being a cloud-based system, it allows the users to share their data with anybody, with collaborators, with anybody. So there's nothing downloadable. There's no programming language that you need to know in order to use it. It's actually fairly straightforward to use. Now, and the other aspect of XEMS Online, which is integrated with the program, is Metlin, which allows us to do the identification. And ultimately, that's in a nutshell, that's largely what we're trying to accomplish doing untargeted metabolomics. Data analysis followed by ultimately the identification of these molecules to pull out the most interesting molecules with respect to a system, whether it's for biomarker analysis or pathway analysis or whatever it is that you're interested in doing. So I'm going to go through a series of stories that I think will give you a sense of how our technology has developed up until the present day, and also where hopefully it's give you a sense of where it's going in the future. One of the first stories that we were involved in was back in 1994. It was involved with sleep, where we were looking at cerebral spinal fluid in sleep-deprived animals with the hope of identifying a molecule that would that could actually promote the sleep process. The idea was to identify molecules that were building up as a result of sleep deprivation and identify them and see if they could be involved in this sleep process. Fairly simple concept, and ultimately we did observe a family of molecules that looked quite interesting. We went through the entire process of the identification with tandem mass spectrometry analysis, accurate mass measurements, HD exchange, isotope pattern analysis, PrEP/LC for the collection of the metabolites for NMR, elemental composition of the MS/MS and the MS-3 fragment ions, and ultimately synthesizing authentic standards to ultimately test the molecule for its activity. And ultimately what we did find was that the molecule, this particular molecule, Olamide, induced the sleep-like state within about 4 minutes of its being introduced to the animal. So it did have some sleep-like properties. From a biological perspective, that of course was particularly interesting. For me though, however, from a technology perspective, what was really disconcerting was that it took us about 8 months to identify this molecule. It was just an incredibly long amount of time, at least for me, as I was just starting to get involved in this area, to ultimately figure out what this was. So it ultimately kept me away from metabolomics for quite a few years. However, what it did do was it actually seeded my thoughts on how to potentially solve this problem. And the simplest way that I could think about it was essentially collect standards that we could then use in a database that would allow us to compare the data that we're getting in these metabolomic experiments and identify them by direct comparison. So a relatively simple concept, and since that original concept was implemented in the early 2000s, we now have over 1 million molecules in Metlin, and more significantly, and I think this is much more significant than just collection of molecules, we now have, actually as of yesterday, I was talking to Winnie Heim, who's been collecting some of this data. We now have over 75,000 standards for which we have MSMS data incorporated into Metlin. So now it's possible within seconds to identify these molecules and many other metabolites and other exogenous molecules in the metabolomics experiment. So, and I should point out that Metlin is freely available. Anybody can access it and look at the data and actually do comparative analysis to identify these compounds. And one of the more exciting aspects of Metlin, and I'm going to spend a few more seconds on it because I do think it's really, at least in our lab, it's been one of the more significant developments over the last roughly 6 to 9 months, is that we've been getting a huge number of standards to incorporate within Metlin. And basically what you're seeing here in these plates represents about 100,000 molecules. Actually, it's a little more than 100,000. It's 105,000 molecules. And what we've been doing is basically taking those molecules, and last year at this time, we had about 15,000 molecules with standards with MSMS data. That number obviously has jumped by over 60,000 in the course of roughly a year. So and the reason why it's jumped so significantly is we've ultimately been able to solve some major problems in the identification process. One of them has been getting standards. This is, again, not a trivial process, actually getting ultimately the hundreds of thousands of molecules that I wanted to get in order to populate Metlin to make it more relevant for solving metabolomics type of problems. But when you get that many molecules, the next question is, how do you prepare these molecules? I mean, you can't do this manually. It has to be an automated process that has to be, if you want to have significant numbers, increases in the numbers where Metlin is being populated by these molecules, you have to have a standardized but automated preparation procedures. And these are a couple of things that we've been able to accomplish recently. The other is ultimately, how do you take this information if you're doing analyzing so many molecules and autonomously transfer that information to the mass spectrometer so it can actually perform the analysis in an automated sort of way. We've been able to accomplish this recently as well. And actually, what's the foundation of everything is the reliable and accurate MS data acquisition of these of the standards. And that may sound trivial, but we've actually gone through about 5 different distinct mass spec ionization type of technologies before we've actually been able to hit on a combination of a couple that are allowing us to acquire the data in a reproducible and very what we believe is accurate and with a very high signal-to-noise ratio capability. And ultimately, the final thing that's been quite laborious is trying to figure out how to stream the data directly into Metlin autonomously. And I'd have to say at this point, the process is still semi-autonomous. It's not as streamlined as it could be, but this is the final obstacle that we're trying to overcome. But we are getting the data into Metlin, and that's taking place in a fairly straightforward manner. What are the reasons why we're doing this? Well, the obvious one is metabolite identification, but there's some other reasons that I think are actually even more compelling as to why we're doing this beyond just simple metabolite identification. One is unknown characterization. If we have so many more molecules as we do right now, we can start doing the similarity searching against the unknowns, and it gets us closer for your unknown characterization than you would have otherwise if you only had about 15,000 molecules in the database. Metabolite annotation. Obviously, people have been working on this for over a decade, and we believe that with so many molecules standards, the annotation process actually becomes much more straightforward. And ultimately, this is something that we're developing within XEMS. Actually, this is being developed by Xavi Domingo, and what he's calling it is XEMS Autonomous Visualization Interface, which we are incorporating into XEMS. And it's also worth noting that with these molecules, we not only have MSMS data, but we also have retention time information as well, which is going to be key, not only for the development of the Xavi algorithm, but it's also going to be key in terms of in silico algorithm development, also for unknown identifications. So again, with this large population of molecules, it'll make it much more straightforward to do this sort of analysis. And I should point out there's been a lot of individuals who have been involved with this. Winnie Heim has been one of the prime movers in this, and I really want to give her thanks for even this weekend. It was a long holiday weekend here in the United States, and she was here every day this weekend collecting data. So I really appreciate her efforts on this for just not only for our lab, but also for the community in general. Okay, the next story I'm going to quickly talk about is stem cell differentiation, which is a story that was developed by Oscar Yanez. Again, it was a fairly straightforward set of experiments where Oscar looked at stem cell differentiation, moving to both cardiac cells and also neurons. For me, one of the more interesting observations that he made was that stem cells would generally contain much more highly unsaturated molecules. It was a very interesting observation because ultimately, when you think about it, it makes a lot of sense. Stem cells containing highly unsaturated molecules that could act as scaffolds for other molecules that would ultimately build up the mature differentiated cells. Made a lot of sense, and one of the key ones that we observed was arachidonic acid. But even more interesting, at least from my perspective, was the fact that he also observed molecules, again, another highly unsaturated molecules, neuroprotectin D1, that when he tested it, it actually increased the differentiation process by roughly 1,500%. So a really significant improvement in the differentiation process just as a result of this one particular molecule. What ultimately came out of this in terms of our technology development, and that was, I would say at the time, we were only focused on XEMS. That's the primary technology, the downloadable version. And what we realized is that we needed to make it easier for a broader population of individuals to use, and that's where the XEMS Online came into existence. Again, through these, where we were seeing a lot of non-experts who wanted to use the technology. Some of the advantages of XEMS Online as a cloud-based system is that it allows for both global and local sharing. You not only if you created data and the results, you can share it easily with your collaborators. All they have to have is access to a computer, and they can look directly at the raw data and also the results, and they can actually reanalyze it themselves if you choose. And there's been actually hundreds of thousands of instances of individuals sharing the data. So it's even though the population of users is on the order of over 20,000, the amount of sharing that goes on is really quite significant. The infrastructure is all housed within at Scripps, so basically you don't have to have any significant infrastructure. Basically, there's no investment on your part, and basically can all be done through our servers. And the other major aspect of this is that you can actually incorporate different aspects of technology into the cloud-based system. That's really very straightforward. I mean, that includes Metlin, it includes the recent pathway analysis and the systems biology capabilities that we have within XCMS Online. So it's really many of the advantages that you get out of the cloud, you get out of XCMS Online. And I just want to mention that Paul Benton is the prime mover behind all of the things that I just mentioned. I would say that well over 90% of the code that's gone into this has come out of Paul's working on this. So I really do appreciate Paul's efforts in all of these different aspects of XCMS. One of which is the pathway analysis, which I just really want to talk about very briefly. It was recently incorporated into XCMS Online, and basically it was developed based on a program that Siuzda Lee invented in 2013, and it was in BMC Bioinformatics, published there, and we incorporated this into XCMS Online, and now it allows us to take raw data from untargeted metabolomic experiments, and in cases like what you're typically used to looking at, which is the box whisker plot, the EIC, the mass spectral data, the Metlin ID. Instead of looking at that, what Siuzda made it us capable of doing, and I'd like to throw up this picture simply because it's most of the collaborators I deal with don't want to see any of this information. What they want to see is what are the pathways that are ultimately being perturbed. And we've actually accomplished this through the incorporation of Siuzda's algorithms and also some of the modifications that we've made in them to ultimately connect out the metabolites directly to pathways. And again, thanks to Siuzda for these developments. The next story is on xenoestrogens or endocrine disruptors that was developed by Ben Worth, who was visiting at our institution last year at Scripps. This was one of a couple of stories that came out of his visit, and this is a cover article, and it was created by Ben. He did a wonderful job on this, and the story on this is focused in on a newly developed drugs combination therapy that came out of Pfizer. It's the palbociclib and the lesotrol combination therapy that Pfizer developed that's been highly successful in treating breast cancer. The question that we asked is, are xenoestrogens, could they possibly have an impact on this combination therapy? A fairly simple question, but given that we're looking at estrogen receptor type of therapy, and the fact that xenoestrogens can interact with those receptors, the question we had would these xenoestrogens available in plant and food material potentially impact this particular treatment? So we used our XCMS-based technology with Metlin, and we used MCSF7 cells to ultimately do these comparative analyses where we treated the MCSF7 cells with the palbociclib and also the lesotrol by themselves. And actually, in this particular case, this is some of the data that we got out of this, and you can see by a quick glance that it's not that significant. The molecules by themselves were not having a huge impact metabolically on the MCSF7 cells, which, if you look at the combination of the two, actually it really highlights the fact that the combination therapy is, at least from a metabolic perspective, is quite effective, which coincides quite nicely with the clinical positive results that Pfizer has obtained with this combination therapy. So from a drug perspective, the results that we've gotten actually have been quite consistent with what the results that Pfizer has obtained from a clinical perspective. So our question was, would xenoestrogens possibly disrupt this activity? And so again, looking at the xenoestrogens with respect to the palbociclib and the lesotrol, we were indeed seeing a significant impact of both the zearalenone xenoestrogen as well as the genistein. Both of them were having a very significant impact on the metabolism that was taking place over the combination therapy. So again, the take-home result, the take-home from this is that if you are using this combination therapy, it may not be such a bad idea to limit your exposure to these xenoestrogens to help the therapy actually proceed in its desired course. Actually, the thing that at least the more interesting thing that we took away from this was that at the same time we were developing a technology with IBM Watson. And as you can imagine, at any one time when we do these metabolomic studies, especially with the xenoestrogens and the exposure type studies that we were interested in, is it possible for us to come up with a streamlined approach to analyzing this data beyond just the manual interpretation that we're used to doing? And this is where IBM Watson came in. What we did was we developed what we call the Watson training approach, where ultimately what we wanted to do was use Watson's ability to mine the literature with known molecules. We trained Watson through an examination of mixture of known molecules that had these exposure effects. This is what we then called Watson primed, which was ready to examine our experimental output. And ultimately, the output is our metabolomics data. And then we used Watson to evaluate these molecules and ultimately prioritize which ones were most significant with respect to, again, this was more of an exposure type of research problem. And the results actually were quite interesting. I mean, we were ultimately looking, in any of these experiments, we can look at hundreds to thousands of potential molecules that are involved in the experiments that we're analyzing. And what Watson did was it actually was able to rank these molecules across roughly 300,000 papers. And perhaps the most interesting part of this is that Watson was able to do this in roughly 4 hours of processing. I should make it clear that it was primarily looking at the abstract, so it wasn't going through all 300,000 papers and every detail within the paper, but it was focusing on the abstracts. But the amount of time it took, I thought, was really quite extraordinary. A very, very short period of time to ultimately prioritize a fairly large set of molecules. And we are actually continuing that collaboration with Watson now. Actually, a very recent study has involved multiple sclerosis, and this is work that we've been doing with Luke Larson, and it resulted in this 2018 paper. It's based on a paper that came out of Luke's lab back in 2013, which is based on the idea that precursor oligodendrocytes, when they differentiate to oligodendrocytes, can ultimately help in the regeneration of the myelin sheath in patients with multiple sclerosis. And what Luke found in this Nature paper was that using a particular molecule, meconazole, a known antifungal drug, it actually repurposed the drug to facilitate the differentiation process of the precursor oligodendrocytes going to oligodendrocytes. And as a result, they saw a significant improvement in myelin sheath regeneration. We decided to look at the same process using metabolomics, looking at precursors going to oligodendrocytes, and then try to observe which of these molecules could potentially have an impact on the differentiation process. Again, XEMS Online and Metlin were used for this, and we ultimately identified one particular molecule of the on the order of 50 or so that we were observing that ultimately had a significant impact on the regeneration of the myelin sheath. Here's the oligodendrocytes without taurine. This is just with meconazole, and this is the oligodendrocytes with the taurine and the meconazole. And what we ultimately saw was a 250% increase in the formation of the oligodendrocytes. And this had a correspondingly significant impact on myelin sheath neuron regeneration. And the results that haven't been published yet, coming out of Luke's lab, is that we've also been able to observe in vivo the impact of taurine on neuron regeneration. So this is not simply an in vitro set of experiments. This has now been reproduced in vivo. So it's again, this endogenous molecule is clearly having a significant impact on the system beyond just the known drug molecule. Part of what came out of that study is the implementation of systems biology within XEMS Online. Basically, now we've allowed it to incorporate proteomics and genomic data to further bolster up the information that we get from the metabolomic pathway analysis. So this partly came as a result of some of this multiple sclerosis work that we were doing, where we saw the need to make it more broad in terms of encompassing other omic technologies. The next study I'm going to talk about, obviously I am talking about a fair number of different types of studies here, but I do want you to keep in mind that I am going to ultimately bring it home with one primary take-home message, a message that I think is particularly important for young investigators in metabolomic type technologies. The study that actually Johannes and I were talking about this prior to me giving this webinar, and it's related to something that he's very interested in particular. The colorectal cancer study was the one that I was interested in. I believe Johannes was interested in pancreatic cancer. But regardless, what we did in these particular studies was we looked at normal tissue. We also looked at left-sided versus right-sided colon and sigmoidal tissue and cecum tissue, and looked at cancers on each side. And we did this in collaboration with David Elder at Karolinska, Cynthia Sayers at Johns Hopkins, Caroline Johnson, who's now at Yale University, and also Laura Getz, who's at UCSD. And again, looking at the up and down regulated molecules, we did see a set of molecules that were particularly interesting. And this is where Johannes comes in, because in his experiments, he sees the same set of molecules. These polyamine molecules are very commonly dysregulated, not only in colorectal cancer, but also across pancreatic cancer. In fact, there's roughly 10 different cancers that see polyamines as being significantly dysregulated in these metabolomic type of experiments. So Johannes obviously finds this interesting from a biomarker perspective, as I do. But what I was thinking about when I started, after we finished the study, the paper came out in 2015, and I started wondering, why is it that these polyamines, the same polyamines, are showing up consistently across multiple different cancers? And of course, they're showing up at different levels. Some very high, like in blood, as in pancreatic cancer, it's a 5-fold difference. Colorectal cancer, it's on the order of a 10-fold difference. But with various types of cancer, you see different levels of these polyamines increasing. So my thought was that perhaps, and this has been done in conjunction with Rafa Montenegro and also Carlos Gijas, who are currently postdocs in my lab. The idea was that perhaps these polyamines and their role is far beyond just the fact that they're biomarkers, and perhaps they're playing a role in immunomodulation. And I have to say, this is a somewhat speculative part of my talk right now, but I think that the data so far that we've generated from this has actually been fairly compelling. So what we've done is we've taken polyamines, a specific set of polyamines, and exposed them to macrophages. And what we are seeing is that the macrophages do become activated. That in itself, I find quite interesting, especially from the immunomodulation perspective. But perhaps what's even more interesting is we are seeing a set of unknown lipids, actually specifically 2 unknown lipids that are being generated from the macrophages as a result of the stimulation with the polyamines. And what even makes that more interesting is the fact that the structure of these molecules, they're complete unknowns. However, their structure they have some striking similarities to S1P, which is of course a known an important immunomodulator. So when I said that this story that we're developing with respect to the polyamines is somewhat speculative at this point, it's speculative because we are now working to synthesize enough of these, what was originally unknown lipids, to ultimately see what kind of, if they have a significant immunomodulation impact. So this is an ongoing story that's taking place in our lab, and I'm sure we'll be developing it significantly further in the near future. And most recently in the lab, I just thought I would throw this in because this is work that we're doing with respect to the microbiome and irritable bowel disease with Professor Dennis Wohl and also Rafa Montenegro here at Scripps. We've been looking at a mouse model for of IBD. And this is very recent data, so it's something we're still developing. But we've observed a relatively small set of molecules that we've been testing in cell-based assays, and ultimately, what we're finding is that 4 of the metabolites actually suppress inflammation, and 7 of the metabolites release intracellular calcium in macrophages. So the activity of these molecules is actually quite compelling in terms of what we're observing with respect to IBD and their subsequent characterization of their activity. Okay, so I did mention that I was going to try to tie this together, and I think the primary thing that I'm trying to bring across in this presentation is not technological, even though I've focused quite a bit on the technology. It's more biological. In pretty much every example that I've given you today, with respect to the metabolites that we've observed, with respect to the polyamines, we've seen some very interesting immunomodulatory activity. With respect to sleep, we saw sleep-inducing properties. Stem cells, we saw the neuroprotectin D1 stimulate the differentiation process. With respect to the xenoestrogens, we saw the impact they were having on the breast cancer palbociclib, lesotrol combination therapy. And with the neurons, we were seeing taurine actually stimulating oligodendrocytes formation, ultimately for its impact on myelin sheath repair. So the activity has been the key theme across all of this, and I think the important take-home message behind this is that this is one of the key things that really distinguishes our omic technology, metabolomics, from the other omic technologies. And when I say it distinguishes it because endogenous metabolites, unlike other approaches, whether you're looking at proteins or whether you're looking at genetic material, the one thing that's really interesting about endogenous metabolites is that they're generally inexpensive to create. Many of the molecules that we do, we analyze and we observe in these studies can easily be purchased out of the Sigma catalog, in some cases for tens of dollars, or as supplements. In fact, at the GNC, for instance, I mean, a lot of these metabolites are very readily available in very high quantities. Taurine is a great example of that because taurine is available in an energy drink called Red Bull. The fact that they're endogenous means that they're generally safe for the most part. Again, using the taurine example in Red Bull, the reason why it's in an energy drink is because it's generally safe. It's an endogenous metabolite. And I think, again, the key fact here is that these two are nice features, but the real interesting part about this is that instead of observing something like biomarkers or pathways, we actually can use these molecules to modulate phenotype. Okay, so is there any precedence for this? I mean, can I point to any studies other than the ones that I've just described that actually show that you can impact a system by introducing metabolites? And of course, the ones that I've described today are all listed here, but there's actually a number of different studies, and this is just this very small list. Actually, there's many more papers out there that describe how you can use endogenous metabolites to impact phenotype. One of my favorites is right in the middle here, the type 2 diabetes example that came out of Harvard and Salk with a colleague of mine, Alan Cyclotarian, who's now at Salk, where actually these fatty acid esters were having a significant impact on insulin resistance. And again, these are endogenous metabolites, so they're ultimately going to be relatively safe. Another really cool example is one molecule, a taconate molecule at the very bottom, and this came out in two separate Nature papers in the last month or so, actually in March and also in April, and from two separate labs. So this is another very interesting molecule with respect to its anti-inflammatory capabilities. Okay, so the primary message is one, that we typically think of metabolites reflecting the phenotype because it's the most downstream approach. And these ideas I've been currently developing with Juliane Banoviskovic and Martin Guerra out of Leiden University. But the reality is that, of course, metabolites are being produced as a result of the DNA, RNA, and proteins. However, the metabolites have an upstream impact on these molecules with respect to gene expression, signal transduction and control of transcription, epigenetic regulation of cofactors, with respect to RNA metabolism, ribo switches and post-translational modifications, and with respect to protein activity, allosteric regulation, or catalysis by cofactors substrates, or post-translational modifications. So what we're trying to use metabolomics for is to ultimately identify the molecules that are involved in these upstream events that can then be used to modulate phenotype. So again, the primary take-home message is coming from that original paper that I mentioned at the very beginning of the talk, is that metabolomics has traditionally been focused on biomarkers. And that's not a distinguishing feature of metabolomics. This is something that we do in transcriptomics. It's something that we do in proteomics, so it's not a distinguishing feature. And the same is true with pathways. Identifying active pathways is not a distinguishing feature of metabolomics. We do this quite often with respect to the other omic technologies. So these are largely passive observations that you can make and try to understand the system that you're analyzing. What I'm proposing instead is actually that we become active participants in the system. Use the molecules that we're observing to see if we can impact the system like we did with taurine or like we did with neuroprotectin D1. So we can ultimately modulate the phenotype, and actually from a metabolomic perspective, we can provide the biomarker information, we can provide the pathway information, but we can take it one significant step beyond that and actually start working with our collaborators to see if we can promote what we think is a positive or an impactful have an impact on the system that we're analyzing. So with that, I'd like to acknowledge some of my funding sources, the NIH, the DoD, the Department of Energy for supporting some of this research. And of course, I've mentioned a number of people that have been involved in this research. And there's many more who have been involved, but I'm providing the key individuals here. And I'd also particularly like to thank Richard Lerner, who originally got me involved in metabolomics before it was even called metabolomics, in that original sleep study that I described at the very beginning. And the last acknowledgement would be to our work with Scott Spangler and Richard Martin at IBM Watson for actually working with us so closely and continuing to work with us so closely to develop this AI technology with respect to the metabolomics output. So with that, I'll be happy to answer any questions that you might have.
Well, thank you very much, Gary. That was really a fascinating talk, and I certainly agree with you, and I think you showed this quite well that metabolites themselves really can be direct effectors, and not simply just bystanders of our molecular machinery, and that they do have the capacity to modulate phenotypes. And I think you gave some really nice examples of that, and I certainly agree with you quite heavily in that regard. So we do have a few questions. These probably relate a little bit more to XEMS or technology, but nonetheless, there is some interest. So one of the questions was actually related to the pathway analysis in XEMS. So it seems that one of our participants had used XEMS using some raw data derived from plant-based metabolomics, and they did not obtain the pathway analysis. So I was wondering, perhaps you could share a little bit of light as to how to go about getting that specific type of data.
Yeah, so the pathway analysis is on the order of, I think, 30 or 40 different organisms are now incorporated into XEMS for pathway analysis. Given the limited information that's available with respect to plant pathways, you can imagine that we can't incorporate into XEMS information that simply doesn't exist. So what we're primarily limited to is incorporating pathways that are known, as opposed to coming up with we're not pulling out completely unknown pathways with respect to XEMS. It's based on data that's been input from other sources to allow us to correlate the molecules that we're seeing with the known pathways.
Okay, thank you. The other questions I had, so one, obviously you've done a great deal of work, particularly with respect to Metlin and getting the MSMS data for a large variety of standards, which I think is very, very valuable, particularly to a lot of us in the field. So one question, though, is, obviously when you run standards, and you mentioned this yourself, there's a big dependency, particularly on retention times. I mean, people have different instruments, different assays that they may run. Is it actually possible to get the standards that you have generated in terms of the plates?
I guess actually the question is, can we provide the standards to the individuals to analyze themselves? Unfortunately, the amount of material that we're getting with that recent plate that I just showed you on the order of 100,000 molecules, there's only on the order of hundreds of femtomoles, if not roughly a picomole, maybe a little bit more than a picomole in some of the molecules. So we're generating data off of our incredibly small amount of material. So unfortunately it's not near enough for me to share with others. It's just basically for us, in many respects, it's a one-shot deal for us to generate that data. Once that material is used, it's essentially gone. So unfortunately, I don't have the capacity to send 100,000 molecules out to anybody who asks for it. It's been really quite challenging for me to get these molecules just to get them into my lab. Being able to share them would be it's unfortunately something we're not capable of doing.
Sure. Understandably. I figured that was a little bit of a long shot. One question though, and I apologize if I mispronounce this, the Xavi. Xavi, thank you. Do you have any sort of a timeline with respect to its implementation? And could you just kind of remind the audience of some of the features that are associated with this new technology?
Yeah, so Xavi, which Xavi Domingo is developing, annotation is not simply just based on MSMS data. Annotation involves many different things. I mean, we're talking about cationization, in-source fragmentation. So the retention time information. So some of the things that Xavi has been incorporating within this Xavi program is to allow us to identify the metabolites that are real and not associated with in-source fragmentation, that are not associated with, of course, just result of redundancy coming from the cationization, and also use things like retention time with respect to these different, the retention time information that we have with respect to all of these different types of molecules to narrow in on identifications that are extremely solid. With respect to the timeline for its development, I believe that Xavi is actually in the final, Xavi Domingo is in the final stages of its development. So maybe it's optimistic, but I'd like to say within about 2 months that the Xavi program would be available on XEMS Online. But I would have to ask Xavi himself where it stands right now. But that being said, I have to say I'm very encouraged by his progress so far, but also the progress that we've made in collecting enough data. And when I say collecting enough data, the data is so far in terms of having standards from which we could generate all of this different types of information. It's actually, I think it's somewhat unprecedented for this sort of information to be available, and unprecedented also from the perspective of anybody who wants to access Metlin can go on and actually use the data. It's of course completely freely available, and I always like to remind people of that. It's not downloadable because we haven't been able to make it downloadable simply because we haven't gotten that sort of funding from the government to actually do that. But what we are doing is making it freely available through the website.
Oh, excellent, thank you. And I think we certainly look forward to that. So a couple more questions. So one of our users said that they had used Metlin, but sometimes they did not find the MSMS spectra, or that the MSMS spectra that they had did not coincide with what was shown in Metlin. And they were wondering how confident they could be of their correct identification, whether or not this may be due to instrument or something else.
Yeah, so what we find is that we've looked at the MS data, in our lab, we have multiple different types of QTOF instruments and, of course, triple quads. So we have QTOF instruments from Agilent, from Waters, from Bruker, from SciEx. And what we generally find is that the MS data is quite consistent across these different instruments. We see good agreement with the Q-Exactive technology, although that's where we may see a little bit more variation, but it's not overly, it doesn't overly vary across any of the instrument platforms. What we do see the variation is in terms of the energy that you're, the quote unquote energy that you're putting into the fragmentation voltage when you do these experiments. Across the different instrument platforms, so for instance, 40 volts on one instrument could ultimately translate into 20 volts on another system. So that's something to keep in mind when you're doing this, that the voltage overlap across the different platforms will vary a little bit, but in general, the fragmentation is quite consistent. So my answer to his or her question is, if they are seeing a significant variation from what's available on Metlin, I think that's a strong indication that they may want to rethink about whether the identity is correct. I've certainly been guilty of incorrectly identifying molecules when I thought the MS data was close enough, and ultimately I was absolutely wrong. So what I would recommend is that in many cases, these molecules are incredibly important because they're going to cause your collaborator to go off and study them for a really significant amount of time. By the standard, perform the experimental and the standard MS analysis side by side, and see how well they match, and also do the LC-MS experiment as well to make sure the retention times actually match as well. So. Yeah, the months or even sometimes years of effort that can go into looking at the biological activity of these molecules certainly justifies purchasing these molecules and at the front end and making sure that you're providing your collaborators with accurate information about the identity.
Thank you, and yeah, I have to admit, I completely agree with your point on that, and I give you resounding support on that answer, because again, I certainly agree. Okay, 2 more questions, and then I think we're about out of time. So one of the questions posed was, is it correct that XCMS Online is not able to process MS data and relate this to the online available in the database? And if so, will this be implemented for metabolite and identification at one stage?
Yeah, so we would love to have that implemented, but we're working on so many different things. Actually, ultimately, where I'd be incredibly excited to have this implemented is in our ability to do this with respect to pathway analysis, because it would make I think based on just the M over Z data that we're putting into the pathway analysis, it does make the pathway analysis more speculative as opposed to sound. So my ultimate goal is to incorporate within what we call an autonomous metabolomics workflow within XCMS, is the XCMS Online, is the ability to have MS data integrated with respect to Metlin to get more solid IDs. But there's beyond with Chavi being developed and some of the other things that we're developing right now, we have to prioritize the things that are at least currently doable, and that is something that we're looking at, but it may not be available in the short term. Okay. But I think that whoever asked that question here, it's a great question, and it's something that I think we should even increase our prioritization of it, because it would be beautiful to have it. The other issue that we're also facing is that with so many molecules now with MS data in Metlin, we have to become a little bit concerned about data processing time and having infrastructure to accommodate all these different types of studies. So there's multiple issues that we're trying to address simultaneously. But it's a great question.
All right, thank you. And last question, and again, this kind of goes back because you had mentioned the ability to use retention time information, particularly as it pertains to identification. So one of the questions was simply, what is the best place to find LC-MS methods that would be amendable, particularly to the data that you have generated, so that others can essentially match the retention times and so forth?
Yeah, I mean, perhaps the best. The papers that I showed at the very beginning of this presentation, if you just Google Scholar my name and look at 2018, there's a number of papers that will provide that information to you. Like the Nature Chemical Biology paper with the multiple sclerosis study is a really good one. There's the work done by Benedict Worth on the xenoestrogens. That's another good source of information. And if you look up Caroline Johnson, who I've had, I think perhaps more papers with her than anybody else. She has a series of papers that we've worked on that really outline the approaches that we use pretty accurately. So I would refer to those papers and those individuals to get that information.
Excellent. Well, thank you again, Gary. This was really an excellent webinar, and we were very appreciative of you taking time out of your day to give it to us. So with that, this will conclude this month's webinar. We certainly do encourage all the attendees to be on the lookout for next month's webinar. We will post this as soon as all the results are finalized. And again, thank you very much, Professor Siuzda. That was an excellent talk, and I certainly believe that it was beneficial to all the attendees.
Okay, thank you very much, Johannes.
Right. Thanks. Have a good day, afternoon, or evening, and we will hopefully see you in the future.